Okay, hello everybody. I'm going to talk to you a little bit about the evolution of novelty today for this lecture, which is part of Advanced Evolution at the University of Idaho. And the key question that I want to ask for this lecture is, how does a novel character evolve? And we're going to focus especially on complicated characters. We can start out with a great example of a complicated character that's come up many times in the history of evolutionary biology, and that's the eye. If you think about your eyes, they're quite complicated structures with many parts, and you can imagine that there's some mystery about how a structure as complicated as an eye might have evolved. Here's a picture that I took from the sort of creationist part of the web. This is a common meme, I guess, that's out there in the world, this quote from Darwin that says, "To suppose that the eye, with all its inimitable contrivances, could have been formed by natural selections, evolution, seems, I freely confess, absurd in the highest degree." And you see this quote, which is a real Darwin quote, a lot in certain circles. I guess implying that even Darwin thought that it was impossible to evolve eyes. The interesting thing about this quote is that it's taken completely out of context. So if we look at the actual quote from Darwin, you can see that the beginning part of the quote is all here. "I freely confess, absurd in the highest degree." But it's followed by this whole longer paragraph that I think is worth reading aloud. "Yet reason tells me that if numerous gradations from a perfect and complex eye to one very imperfect and simple, each grade being useful to its possessor, can be shown to exist, if further the eye does vary ever so slightly and the variations be inherited, which is certainly the case, and if any variation or modification in the organ be ever useful to an animal under changing conditions of life, then the difficulty of believing that a perfect and complex eye could be formed by natural selection, though insuperable by our imagination, can hardly be considered real. How a nerve comes to be sensitive to light hardly concerns us more than how life itself first originated, but I may remark that several facts make me suspect that any sensitive nerve may be rendered sensitive to light, and likewise to those coarser vibrations of the air which produce sound." Now, there's a couple things that are worth noting here. One, that Darwin actually comes up with a plausible mechanism for the evolution of the eye, following this quote that people often pull from The Origin of Species. Two, you can tell from this quote that Darwin really has no clue how eyes actually evolved. And now we know quite a lot about the evolution of the eye, and so we can fill in some of Darwin's mysteries a lot more closely. And third, Darwin has a gradualistic view of the origin of the eye, which turns out to not be completely accurate. So let's delve into that a little bit. Based on a lot of research that's happened in the last 30 years or so, we now have a really good idea of how the eye evolved. And it turns out that this picture of how the eye evolved gives us really deep insights into some general features about the evolution of novelty. We can summarize those things that we've learned from the evolution of the eye and other complex characters in this way. We can contrast the traditional perspective, which is kind of what Darwin described, a simple origin of some light-sensitive neuron, and then gradual elaboration of that structure with each step being beneficial to the organism. I do want to say before I go on that this traditional perspective on simple origins and gradual elaboration does apply to the evolution of some complex characters, but not really well to the eye. A better perspective on how the eye itself evolved, and I think applies to other complex characters as well, is that the eye evolved through a process of co-option and tinkering. And I'll talk more about tinkering later in the lecture, but also in the next lecture in this series. So let's go into a little bit of detail about the evolution of the eye so we can get a picture of this ways of co-option and tinkering and how they work. And most of the information I'm going to give you now about eye evolution comes from a combination of comparative anatomy and phylogenetics. One of the core ideas that people use to try to reconstruct the origin of complex characters is move from the simple picture that we see in A here of a character evolving suddenly on the tree to a picture that's more nuanced and summarizes how the parts of that character have evolved. So, for example, if we think about the eye with all of its complicated structures, both morphologically and genetically, and we break those down instead of just presence or absence into parts, often what we see is a pattern more like what we have here in C, which is that the different parts of the eye have different evolutionary origins, and then they get reassembled in different combinations to carry out specialized functions in organisms. For example, if we look at the light-sensitive organ itself and how it works, we can trace origins of light-sensitive organs all the way back deep into the evolutionary tree of life by just looking at organisms that are distantly related to humans, like this euglena. And you can see here this little red thing, that's its eye spot. Here's a conceptual diagram of the euglena. You can see the eye spot here. And when light hits that eye spot, it starts what's called a phototransduction cascade that transmits the information that light is present in the environment to a processing system in the organism. And actually, we can look even deeper at these phototransduction cascades themselves. This is like the example of taking a character that's complicated and breaking it down into parts. So one of the parts that's common to all visual systems is phototransduction cascades. And by looking at that character in particular, we can learn quite a lot. In euglena, the phototransduction cascade starts with an enzyme called adenyl cyclase that's activated by blue light. That adenyl cyclase transforms cyclic AMP and starts a signal transduction pathway that can result in a response in the euglena. And those organisms go towards light so they can carry out photosynthesis. If we look at phototransduction cascades in the bilaterian animals, that is the bilaterally symmetrical animals like ourselves and many of our relatives that we're interested in, we can see some really interesting patterns. For example, here is a summary table from a paper from Oakley and colleagues showing 4 different phototransduction cascades across animals spanning from flies to jellyfish. And you can see actually that even within this relatively specialized character of phototransduction cascades, we have different pieces that work in different ways. So the opsin that senses light is different across the groups. The intermediary enzyme that carries that message is different. The mechanism by which the message is carried is different, and the ion channels involved are different. We can then reconstruct the evolution of all of those different pieces of the signal transduction cascade on the tree of animals, and we see some really complicated patterns. Now, this diagram that I have here on the left is really complicated, and I'm not going to go through it in great detail, but I will say a couple things about it. You see 3 different colors there, green, blue, and red. Those 3 colors are 3 different opsins that are involved in the signal transduction cascade across animals. And so you can see for animals that have all of these different opsins present, like a mouse, here's a mouse, here's a mouse, here's a mouse, here's a mouse, you can see that they have multiple copies of these opsins that result from duplication and divergence events deep in evolutionary history. And you can also see that if you look at the different aspects of the signal transduction pathway, there's a lot of variation across species that's in a really complicated pattern. You see that here. There's also a lot of question marks, so we don't completely understand all of these things. But the major features of this figure that I want you to know. 1, we have a deep origin of signal transduction pathways used for vision that happened in the earliest animals. That deep origin didn't involve evolving new genes from scratch, but rather repurposing of even older signal transduction cascades that are used inside cells for lots of different purposes. So basically, these early animals took signal transduction cascades that they already had, connected them to light sensing enzymes, and used that machinery to respond to light. We can also see a complicated pattern of gene duplication and divergence. So genes are duplicating, and then the 2 different duplicates can carry out slightly different tasks. And also we see a complex pattern of evolution that doesn't just go from simple to complex, but rather is much more complicated than that. And we can do a similar sort of analysis by looking at different structures in the eye besides just the signal transduction cascade, and I'll post a YouTube video for you to watch that kind of captures some of these other structures and how they've evolved. The main point of all of this is that at least sometimes, and the eye is a good example of this, novel characters can evolve through the repurposing and reassembly of genomic and morphological components in complicated ways throughout evolutionary history. And this is, I think, a good take-home message that we can learn about the evolution of novelty by studying the eye. Next, I'll talk to you a little bit more about the concept of evolution as a tinkerer and the consequences that has for us.